A very good afternoon to everyone. Welcome in the class. Today we are going to talk about protoplast fusion, cybrid and hybrid. This is the topic of our class. Before moving with the topic, let us have a small introduction about Unacademy. Unacademy is one of the India's largest platform for the preparation of CSIR UGC NET Examination. I'm your educator Farida Bandookwala. I have a teaching experience of 10 years and I've qualified MP-SLET examination and I'm pursuing my PhD from School of Biotechnology DAPV. If you are opting for Unacademy, what all things you are going to get? You are going to get daily life classes, you are going to get a well-structured course, you are going to get live tests and quizzes, and you are going to get unlimited access to all the videos which are recorded in that particular category. Like if you are going for CSIR UGC NET, you'll get unlimited access for all the videos which have once been live, recorded, or anything. If you're watching a video today, you can watch the same video after 4 months if you're going for the subscription. Unacademy tries to provide free live classes as much as possible. These free live classes it gives on YouTube and on the application that is Unacademy Learning app. The free classes which are conducted on Unacademy Learning app are called as special classes. So you can get the benefit of free live classes on YouTube and on special platform. YouTube you are accessing now. Just you can subscribe the channel and you can get the notification for all the upcoming videos. And for special class, what you need to do is you need to download the app from Unacademy from Play Store and need to write Unacademy Learning app. Then you will be directed towards the Unacademy Learning app. Download the app, make your account, and follow my profile by the name Farida Bandookwala and you will be able to access all the videos which I have recorded previously or I am going to in future. Then there is a paid subscription. This paid subscription is called as Plus subscription. There are all the videos which are in the Plus subscription are well structured, formulated according to CSIR net. There are total 13 units and you can get all the 13 units on this platform. And another thing is, despite being a paid platform, it is very pocket friendly. The amount is quite less, that is 11,550 for 12 months and 16,800 for 24 months. Because recently a discount is going on and soon the prices are going to increase. And another thing, if you are using my referral code, you will be getting an additional 10% discount by using my referral code FB01. So do not forget to use my referral code FB01 and do not miss the chance of getting a 10% discount. If you're using my referral code, the amount is going to be much lesser, that is 10,395 and 15,120 rupees. Right, so this was a small introduction about Unacademy, and do not forget to go for the Plus subscription because soon the prices are going to increase. Like from 31st December, prices are going to increase. Recently, a major discount is going on. And another thing that if you're going for Plus subscription, it's not so that you will have the access towards all the videos of my profile. You will get all the videos of all the educators who have done the videos under CSIR UGC NET category or gate category. So do not forget to take the Plus subscription as soon as possible, right? So let us talk about the topic that is Protoplast Fusion. Protoplast Fusion, right? This is the topic. Now, if we are saying protoplast and we are saying fusion, so protoplast is one word and fusion is one word. We know what is the meaning of fusion, that is joining of two things. But if we are saying protoplast, what is a protoplast? So first we'll understand that what is a protoplast and if we have the protoplast, how they can be fused. So what is a protoplast? Protoplast is a plant or bacterial or fungal cell that has its cell wall completely or partially removed using mechanical or enzymatic means. Enzymatic means. This is a protoplast. So what all words we are using over here? It can be a plant material, it can be a bacterial material, or it can be a fungal material, because they have got cell wall. So if cell wall of plant cell, bacterial cell, or fungal cell is removed completely or partially, now if you have to remove the cell wall, it can be a gentle mechanical method, or it could be enzymatic method. So any one of the method, if one is using for removing the cell wall, it is what we are going to get in the end. We are going to get a protoplast. So this is protoplast fusion. So this is the definition. This is the actual definition of protoplast. Now protoplast has got another synonym or another word along with it, which is usually asked or used. So one is protoplast, another is spheroplast. Protoplast and spheroplast. What is a protoplast? Have their cell wall entirely removed. Entirely removed cell wall. And spheroplast partially removed cell wall. Now remember that protoplast and spheroplast. So protoplast, we are saying fusion. So fusion, when fusion will occur in a well systematic manner or successful manner, all the cell wall is removed, right? So remember protoplast fusion, and imagine that successful fusion can occur with a protoplast. So protoplast has entirely removed cell wall and spheroplast has partially removed cell wall. Now, more generally, protoplast refers to the unit of biology which is composed of cell's nucleus and the surrounding protoplasmic material. Right? So, what is this protoplast made up of? Protoplast is made up of Protoplast is made up of nucleus plus surrounding protoplasm. Right? So this is a protoplast made up of nucleus and surrounding protoplasm. Now protoplasm, what is a protoplasm? How it will look like if we are talking about that? Protoplast. So now they don't have cell wall, they lack cell wall. So they are naked spherical cells which are obtained from plants. Obtained from plants by removing cell wall. By removing cell wall and it's cultivated in liquid or solid medium, right? As we are talking about plant tissue culture since yesterday's class, yesterday we did artificial seed. So protoplast, whenever a word comes, protoplast or protoplast fusion, why are we actually obtaining this protoplast? What must be the aim of obtaining a protoplast or growing it in the artificial medium, right? So that is the main purpose when we are talking about protoplast, though there are many other study purposes also, but over here in protoplast fusion we are going to grow it in the liquid or solid medium. So what is a protoplast? It is a naked spherical cells which is obtained by removing the cell wall, and then it can be cultivated in liquid or solid medium, right? Now protoplast are the cells of which uh, cell walls are removed by cytoplasmic and cytoplasmic membrane is the outer membrane. So if we are saying that protoplast is naked, see this word naked, and we are also saying that it is spherical, right? Two words we are saying. So naked we are using for no cell wall, but cell membrane present. But cell membrane present, right? And spherical we are saying spherical. Why we are saying? Because of cell membrane. Because of cell membrane. So these are the two words which we are using over here, right? And because of cell membrane, which is the outermost layer, which is the outermost layer, right? This we are saying. Now if we are saying that we are reading protoplast fusion, so we should know a little bit about it. So this term term protoplasm was actually given by Hanstein, and it was given in year 1880, and after that in year 1892 first isolation of protoplast was done. First isolation, and this first isolation was done by Flecker, and this first isolation was actually a mechanical isolation. Mechanical isolation, right? So these are some of the initial things. Now lastly, in year 1971 also something happened. So in 1971, Rakab et al, they were actually successful to achieve a full viable tobacco plant from the protoplast. So achieved success to get complete tobacco plant from protoplast. From protoplast. So this is about a little bit history of protoplast fusion, right? Now if we are saying protoplast, so the way to isolate the protoplast, that should be seen. So isolation of protoplast, right? Isolation of protoplast. So protoplast, it can be isolated from any part of the plant. So it can be isolated from from roots, leaves, fruits, tubers, root nodules, endosperms, pollen mother cells, etc. Right? So it can be isolated from roots, it can be isolated from leaves, can be isolated from fruits, from tubers, from root nodules, from endosperm, from pollen mother cells. So this is the isolation of protoplast. Now protoplast can be isolated from cells by two methods. Methods of isolation. Methods of isolation. What all are the methods of isolation? Mechanical method and enzymatic method. Mechanical and enzymatic method. Two methods are there. Now we'll talk about enzymatic method. Enzymatic method. So we need to remove the cell wall. So the plant cell wall is actually made up of what? It is made up of, we know, cellulose mostly. Then it is made up of hemicellulose. Then it is made up of pectin. So these can be degraded very easily by cellulase, hemicellulase, and pectinase. So what can be done? In plant cells, we can use cellulase, hemicellulase, and pectinase at around pH of 4.5, pH of 4.5, and temperature 25 to 30 degrees Celsius, and incubation period of 20 hours, and we can obtain the viable protoplast, right? So this is the enzymatic method by usage of enzyme, and we know that cell wall is made up of these things, right? So the enzymes which can be used, these are the enzymes. Now this was the enzymatic method. Let us move towards mechanical method. Mechanical method. So mechanical method, if we are going for mechanical method, we cannot go for a bigger piece. Even in enzymatic method, we cannot go for a very big piece. We have to select some small pieces of epidermis selected. Small pieces of epidermis is selected. Then these cells are subjected to plasmolysis. So because of plasmolysis, the shrinking of protoplast away from the cell wall occurs. So we'll write that plasmolysis, what it will lead into, small pieces of epidermis is selected, subjected to plasmolysis. I should have written that. Subjected to plasmolysis. Subjected to plasmolysis. So protoplast shrinks away from cell wall. Then the tissue is dissected to remove the protoplast. The tissue is dissected to get protoplast. But it's not a gentle method. There are disadvantages associated with it. So what are the disadvantages? The number of protoplasts which is obtained are small number of protoplast obtained. Then it is not suitable for isolating protoplast from the meristematic and less vacuolated cells, and this method is quite laborious and tedious because one has to dissect it, so it is quite laborious and tedious. So like this, we have seen the mechanical method. The most important point which you should remember in this, the words which you should remember actually, is dissected, and plasmolysis. You can remember these two words very easily. Rest of the things will come in your mind. So this was the mechanical method. Now when we have obtained the protoplast, maybe by enzymatic method or mechanical method, we cannot directly go on using them. We need to purify them so that we can actually have a proper protoplast. So the next step is purification. Purification or selection of a viable protoplast. That is the thing. Now, the first method which is used for purification is sedimentation and washing. Sedimentation and washing. So what happens in sedimentation and washing? In this method, the crude protoplast suspension is centrifuged. So protoplast suspension, all the protoplast which we have obtained, that is in the suspension form. So protoplast suspension, it is centrifuged. It is centrifuged at low speed. So this slow speed 50 to 100 gram for 5 minutes. The intact protoplast will form a pellet. That protoplast are going to be heavier than the cell wall which is removed. So intact protoplast form a pellet, and the supernatant containing the cell debris or the broken cell wall, that can be pipetted off. So supernatant contains cell debris plus cell wall, can be pipetted out, can be pipetted out. Now the pellet is resuspended in the fresh culture very gently, and mannitol will be added and it will be rewashed. So pellet is gently resuspended in fresh medium plus mannitol and rewashed. This process is repeated for 2 to 3 times. Repeated 2 to 3 times to get the clean protoplast preparation, and clean protoplast is obtained, right? So this was the first method, that is sedimentation and washing. So we should highlight it. Sedimentation and washing, right? Now the second is flotation method of purifying the protoplast. Flotation. So a concentrated solution of mannitol, sorbitol and sucrose can be used as a gradient. So what all things will be used? Mannitol plus sorbitol plus sucrose, 0.3 to 0.6 molar, and used as a gradient, and crude protoplast suspension and crude protoplast suspension can be centrifuged, at appropriate speed, So the protoplast which are lighter, which are of low density, Then the other debris, they allow the protoplast to float and the cell debris to sediment. So protoplast of low density float, and cell debris, that is the dead cells, cell debris, they sediment. So in this case, protoplast will be obtained in the first layer. So one more step is there. So we'll write over here. Protoplast can be pipetted off from the top after centrifugation. So protoplast can be pipetted out. Can be pipetted out. Right. So this is the flotation method. In this flotation method and in the sedimentation method, both of the methods, the protoplast obtained is at different place. In the washing and sedimentation method, what happened? The protoplast was obtained in the bottom, and here in the flotation method, the protoplast is obtained in the top layer. Right. This is the thing. Now coming to the point, how to fuse them. Right, that is the thing, how to fuse these protoplast. So let me take pen. How to fuse. So method of fusion, method of protoplast fusion. So protoplast fusion, they can be classified into two categories, that is a spontaneous fusion or induced fusion. So we'll write this spontaneous fusion, spontaneous fusion, and induced fusion. Now in spontaneous fusion, what happens that they fuse through the plasmodesmata, fuse through, that is the place where the cells they communicate. So they fuse through their plasmodesmata, and in induced fusion they needs fusing chemical, needs fusing or fusion chemicals. So method of protoplast fusion can be spontaneous or can be induced. Like spontaneous, if they are left out, they'll fuse directly without any outside assistance by their plasmodesmata. And if we are talking about induced fusion, they will need some kind of chemicals to induce this fusion, right? Now the protoplast fusion, they can fall into altogether different category as well. So this is direct and indirect, spontaneous and induced. Now there is one more category of this, that is mechanical fusion. Mechanical fusion, so term itself is telling that it will need some kind of mechanical force. Then after that, chemo fusion, so it will need some kind of chemicals. And electrofusion, and electro fusion, it will need electricity for fusing. So mechanical, chemo and electrofusion, right? These are the things. So we'll discuss first of all spontaneous fusion. Spontaneous fusion. So the term itself is clear that what is spontaneous fusion. So whenever the cells, they'll be subjected to the enzymatic degradation of the cell walls, some of the cells which have lost their protoplast, those who are actually near to each other, can fuse to form a homokaryotes, right, or homokaryons, having the same kind of nucleus, homokaryons. These fused cells may sometimes contain higher number of nuclei because there is some kind of fusion. So because of the expansion and subsequent coalescence of the plasmodesmal connection between the cells. So this is the time when spontaneous fusion occurs. Immediately when the cells are subjected to enzymatic degradation. So spontaneous fusion will occur during enzymatic degradation of cell walls. So some of the adjoining protoplast may fuse. Some of adjoining protoplast some of the adjoining protoplast fuse to form homokaryons, and these fused cells will contain sometimes higher amount of nuclei, may contain higher amount of nuclei because of expansion and subsequent coalescence of the plasmodermal connection between the cells. So these are the three things which occurs in the in the spontaneous fusion. So the reason of higher amount of nuclei can be expansion and subsequent coalescence of plasmodermal connection of plasmodermal connection between cells. I hope you people are understanding. Let me see the live chat section. There is a live chat section. You can anytime comment in this live chat connection. Shona is writing, hello ma'am, I love the way you teach. Thank you Shona for your comment. If you are having any kind of doubt, you can write, and I would really appreciate if you're coming back in every day's class because it is a crash course going on for GATE and CSIR, GATE in the evening and CSIR in the morning. So it is going to be a great help for the students who are actually aiming for these examination. Okay, I can see that Shona is listening the class live. Now let us talk about mechanical fusion. We have seen spontaneous fusion, and so now let me change the page. See, spontaneous fusion, we have seen that it occurs through plasmatasmata. We have seen, and mechanical, chemo and electro, these are actually types of induced fusions, right? Because mechanical fusion, that is also induced. Chemo fusion is also a way of induced fusion, and electrofusion is also a way of induced fusion, right? So we are talking about induced fusions now. In induced fusions, first is mechanical fusion. Mechanical fusion. This is the first fusion which we are seeing. So in this, the isolated protoplasts are brought into intimate physical contact mechanically. So we'll write that isolated protoplasts are brought into intimate physical contact mechanically, because it is a mechanical method, right? So if we are saying mechanically, so what is it? So this will occur under microscope and using micro manipulator or perfusion micropipette. If you have seen the concept of gene gun or gene bombardment, so that kind of thing, that micro manipulator will be used under microscope. It will be performed, and it will be performed using micro manipulator or perfusion pipette, micropipette, that too, because it is a very small fragile structure, so it will need micro manipulator, very small one, right? So this is the mechanical fusion, and it is done using a sophisticated machine, right? So this is the thing. Now when we are talking about next thing, that is chemo fusion. that is Chemo Fusion, that is with the help of chemical. This is the second one, Chemo Fusion. So there are many chemicals which can be used to induce the protoplast fusion. The examples can be sodium nitrite, polyethylene glycol, or calcium ions. These all things can be used. These are the examples of chemicals which can be used. Chemicals which can be used. Right? These are the chemicals. Now we are talking about a very specific chemical, that is sodium nitrite. So how is it going to be? Let us talk about that. So the isolated protoplasts, they are exposed to a mixture of, so isolated protoplast, they are exposed protoplast, they are exposed to a mixture of 55% of sodium nitrite in 10% sucrose solution, and the incubation will be carried out for 5 minutes. 5 minutes, incubation of 5 minutes, temperature at 35 degrees Celsius, and then it will be followed by centrifugation. And the protoplast pellet kept in will be obtained. So that pellet will be kept in the water bath for 30 minutes at 30 degrees Celsius, during which the actual fusion is going to occur. So we'll write this, that protoplast pellet will be kept in water bath, pellet in water bath at 30 degrees Celsius for 30 minutes, and this is the time when fusion occurs. This is the time fusion occurs. So this is the first thing that we have seen, that is sodium nitrite, right? Now we'll move towards the treatment with calcium ions. Treatment with calcium ions at high pH. At high pH. This is the second. So this method is actually composed of incubating the protoplast in a solution of 0.4 molar mannitol containing 0.05 molar calcium chloride, and the pH of the solution should be adjusted to 10.5, and this pH is adjusted with the help of glycine plus NaOH buffer. So this is the composition. So this is the way how calcium ions will be used. So here is the place where we are saying calcium ions. So in this, the incubation of protoplast is going to occur in this solution. So, and the temperature for this would be 37 degrees Celsius, and time of incubation for this is 30 to 40 minutes. Sorry, the time taken, this is the incubation time, and the fusion and the fusion will take 10 minutes. Fusion will take 10 minutes. So in this whole thing, the protoplasts will be added. Isolated protoplast, I must write, isolated protoplasts are added. So this is the way we perform the treatment with calcium ions. And if we have seen polyethylene glycol, so let us talk about that also. But before that, I need to add some slides in the PPT. So just be there. I'll put the slides and we'll resume back. So I'm adding the slides. Right. So I've added them. And now we need to talk about polyethylene glycol, right? Polyethylene glycol. So what happens in polyethylene glycol? The isolated protoplast in the culture medium, it will be mixed. So isolated protoplasts are mixed in the culture medium, or protoplasts in culture medium. They are mixed with equal volume of 1 ml of, so isolated protoplast in culture medium, that is also 1 ml, and 1 ml of 28 to 56% of polyethylene glycol. They'll be taken in a tube. They'll be taken in a tube, and then the tube is shaken. Tube is shaken and then allowed to settle. So after vigorous shaking, they'll be allowed to settle. So the settled protoplasts which will be obtained, the settled protoplasts, they'll be obtained and they'll be washed. Protoplasts are washed several times, and during this the fusion occurs. This is the method that is done by polyethylene glycol. So these are the very three famous steps, or the three methods which are used for protoplast fusion: chemo fusion, sodium nitrite fusion, and polyethylene glycol fusion. These are the three very famous and highly used methods. Now in the category of induced fusion, we have seen mechanical, we have seen chemical, now we need to see electrofusion, right? So we'll be talking about electrofusion. So this is the category of induced fusion only. So if we are saying electrofusion, obviously some electric charge will be used. So electric field of low strength is used. Electric field of low strength that is 10 kilovolt per minute will be used, and this gives rise to the dielectrophoretic dipole generation within the protoplast suspension, and within this, or within microseconds, the fusion will occur. So let us write that will generate dielectrophoretic dipole, and protoplast fusion occurs. So this is also a matter of chance, that where it is occurring and where it is not occurring, right? So now we have seen how to isolate protoplast, how to fuse them, and now we need to see that when there is a successful protoplast fusion, what all things we are going to get. So let me just check the live chat box. I can see, hello ma'am, I love you the way you teach. Rukaya, she's saying hi ma'am, how are you? Love the way you teach. Crystal clears up. Okay, Shakira is saying hi ma'am. Hello everyone. Thank you to be there in my class. It really motivates us when people, they watch the class and they appreciate. So thank you very much. Now after electrofusion, we are going to talk about what are the fusion products, right? So let us talk about fusion products. What all things we are going to get after protoplast fusion. So fusion products can be hybrids or cybrids, right? So these two words also I have included in the description of the video, that we are going to talk about protoplast fusion, and then we are going to talk about hybrids and cybrids. So fusion of cytoplasm of two protoplasts result in the coalescence of the cytoplasm. The nuclei of two protoplasts may fuse or may not fuse even after the fusion of cytoplasm. So nucleus may fuse or may not fuse, right? So what all things we are going to get? So we are going to get binucleate cells, binucleate cells known as heterokaryon, heterokaryon or heterocytes, heterocytes. And when the nuclei they fuse, nuclei are fused and the cells are obtained, they are called as hybrids. When nuclei fuse, when nuclei fuse, what we get? We get hybrid cells or we get synkaryons. Synkaryons, right? And when only cytoplasms they fuse and genetic information from one of the two nuclei is lost, it is called as cybrid. When nuclei doesn't fuse, but cytoplasm fuse, but cytoplasm fuse called as cytoplasmic hybrids or cybrids. This is the word, right? So cybrids and hybrids, we have covered that what things will be called as cybrid and what thing will be called as hybrid. Now, what is the application of the somatic hybridization and cybridization? The application part should be seen clearly because that is the place where it is applied in the real life. So application. So first application is somatic cell fusion appears to be the only means through which two different parental genomes can be recombined among the plants and can reproduce uh, sexually. So somatic fusion appears to be the only means through which the parental genomes can be recombined among the plants, which cannot actually go for sexual reproduction, right? So somatic cell fusion without sexual reproduction, without sexual reproduction, if fusion of parental genome is possible. Genome is possible. First thing. Then protoplast of sexually sterile plants can be fused to produce fertile diploid polyploids. So protoplasts of sexually sterile plants can be fused to produce fertile diploid diploids or can be polyploids can also be obtained, diploids or polyploids. Now somatic cell fusion overcomes the sexual incompatibility barrier, overcomes sexual incompatibility barriers. In some cases, the somatic hybrids between two incompatible plants have also found the application in industry and in agriculture. Various different type of combination we see these days while we are surfing on the internet. So these all are the results of somatic hybridization. Many disease resistance genes, they can be successfully transferred from one species to another species through this. So many disease resistant genes can be successfully transferred from one species to another. Then the genes responsible for tolerance of cold, frost, and salt could be successfully introduced through somatic hybridization. So, introduction of cold intolerance, not intolerance, cold tolerance genes in tomato is a success through somatic hybridization. So, we'll write one of the example here, that cold tolerance gene was was introduced in tomatoes. In tomatoes. Then somatic hybrids with high nicotine content and low erucic acid have been developed. So another example is of hybrids with high nicotine and low erucic acid is developed, and cybridization has made it possible to transfer the cytoplasmic male sterility. So hybridization hybridization has made possible to transfer cytoplasmic male sterility. So these are some of the great advantages which are linked with it. Now we'll put some light on the limitations, and then we'll sum up limitations. So it does not always produce the plants that give fertile or viable seeds. Non-fertile and non-viable seeds can be a problem. Viable seeds can be a problem. Then regenerated plants which are obtained from somatic hybridization are often variable because of the somaclonal variations, chromosomal eliminations, various things. So many variations are inculcated. Inculcated. Then protoplast culture is associated with genetic instability. So genetic instability is another thing which is associated with it. Then there is no certainty as regards the expression of any specific character that this is going to be expressed. There is no surety. Then there are limitation in selection of hybrids as many of them are not efficient. Then somatic hybridization between two diploids and formation of amphidiploid, which is not favorable. That is why haploid protoplasts are moreover preferred as compared to diploid protoplasts. So these are some of the limitations which are associated with the topic that we have seen. Now let us do a very quick revision of what all things we have seen in today's class. Let me just see who all students are live, and is there any query? So just a second. Yeah, so I'm directed towards the live chat. So we have talked about protoplast fusion. We have seen the definition, then we have seen what is a protoplast and what is a spheroplast. Then we have seen that if we are saying in protoplast, what all things we are talking about. And if we are saying in the definition that it is naked and spherical, so what makes it naked? Because of the absence of cell wall. And what makes it spherical? Because of the presence of cell membrane. So that is a protoplast. Then we have seen a very precise history of how this protoplast fusion came into being. Then we have seen isolation of protoplast. Protoplast can be prepared from roots, leaves, fruits, tubers, root nodules, endosperms, pollen mother cells, etc. can be used for this. Then what are the methods of isolation? Methods of isolation can be mechanical and can be enzymatic. Enzymatic method, we have seen that what all enzymes we can use. In mechanical method, we have seen that how is it performed. That is two important words I asked you to remember. That is plasmolysis and dissected. These two are the keywords. Then purification, two things we have seen, sedimentation and washing, and the second was flotation, right? These two things we have seen. Then what are the methods of protoplast fusion? It can be spontaneous fusion, it can be induced fusion. When we're talking about induced fusion, it is again of three types, that is mechanical, chemo and electro. These things we have seen. And after that, we have seen in chemo, we have seen three types, that is sodium nitrite and calcium ion dependent and polyethylene glycol dependent. So here we have seen sodium nitrite, right? And this one is our calcium treatment. Calcium treatment. So this was our second one, calcium treatment, right? So after this, we have seen Electro Fusion, and after that we have seen that what are the fusion products. Very important, because this is what I've written in the description, that we'll be talking about hybrids and cybrids. So you should know the difference between hybrids and cybrids, which I have made quite clear, that when the nuclei they fuse, they are called as hybrid cells, and when nuclei doesn't fuse, they are called as cytoplasmic hybrids or hybrids, right? Very easy to remember because the words itself are self-explanatory. After that, what is the application? We have seen, so there are various applications of this method, and after application we have seen some of the limitation also, because nothing comes with 100% advantage. There are always some cons related. So this is it for the topic of today. We'll be connecting again tomorrow with some other interesting topic uh, related to the GATE crash course, because GATE crash course is going on for GATE 2021, and again tomorrow at 5 o'clock I'll be meeting you all and we'll be discussing some other interesting topic of GATE. So if you haven't subscribed to our channel, that is Let's Crack CSIR UGC NET till now, please subscribe it, hit the bell icon for getting all the notifications of the upcoming videos. And if you really like my video, please give it a thumbs up. It really motivates the educators like us. And as I told you, a discount is going on in the Plus subscription. Do not forget to use my referral code, that is FP01, to avail a 10% discount. And if you are going for a Plus subscription using my referral code, that is FP01, you will get access towards all the educators videos, not only mine, other educators video also you can see in the category of CSIR UGC NET. So hurry up for the subscription, because prices are soon going to hike. You are planning that I'll do this from the new year, so don't wait for that new year, just do it now, because presently discount is going on and soon the prices are going to increase. And also try to watch my videos on the Unacademy learning app, that is on the play. You can download the app from the Play Store, Unacademy learning app. Make your account and follow my profile by the name Farida Bandookwala, and you will be able to watch my videos over there. You can watch the videos of other educators as well. So this is it for today's class. We'll be meeting tomorrow again. Till then, thank you very much. Have a very good day. Keep on studying and come back tomorrow. If you have any kind of query or you want any specific topic to be taught by me, you can write in the comment box. Whenever I'll be coming back to my video, I'll be definitely taking up your query, and I'll try to make a video on that specific topic which you are asking for. So do not forget to write your queries also, because that tells that yes, you are paying some kind of attention. So this is it for now. We'll be meeting tomorrow again at 5 o'clock and 10:30 in the morning for our CSIR, and evening 5 o'clock for our